So, to get started, I'd like to introduce today's speaker, Tim Martin. Tim is a faculty member in the School of Forest Resources and Conservation at the University of Florida. He has a bachelor's in forest management from Oklahoma State University, a master's in tree physiology from the University of Georgia, and a PhD in tree physiology from the University of Washington. His research focuses on carbon, water, and nutrient relations in southern pine plantations, and he teaches undergraduate and graduate courses in forestry and tree physiology. Tim?
Thanks, Sean. I appreciate everybody being here this afternoon, and it'll be interesting giving a lecture to a class full of people that are all muted. I need to look into having that implemented in my classes here at UF. It sounds like an interesting way to go. So thanks everybody for being here. And if you do have any questions, please feel free to put them in the chat box there, and I'll be happy to attempt to answer those as we go. So as Sean mentioned, we're going to continue today what Mike Tyree started yesterday. Mike gave a really nice summary of the biology and ecology of water and water stress in trees and forests. I'm going to continue along that theme with a little bit of information about the response of trees and forest to temperature. So I'll talk a little bit about that, and then I'll move on into management and spend some time talking about how to manage for temperature and how to manage for water, either too much water or not enough water in forests. So that's a general idea of what we're going to be talking about today. So let's jump right into temperature. When we're thinking about temperature response and the biology of that response, one place to start is thinking about how photosynthesis responds to temperature. And this is a graph showing photosynthesis of different species versus temperature. And what we have are species from the temperate zone right here, and an example of a couple of species from the tropics as well. And what you'll see is that the shape is the same. Photosynthesis has a temperature optimum where it's the highest, and then it tends to fall away at temperatures below the optimum or above the optimum. And those changes occur for different reasons. So below the temperature optimum, those reductions in photosynthesis tend to be because the enzymatic response of the reactions that make up photosynthesis just go slower at those slower temperatures, and that tends to limit the rate of photosynthesis. Now, above the temperature optimum, we tend to see a fall off in this net photosynthesis rate for a different reason, and that's because as temperature rises, the rate of respiration rises faster than the rate of gross photosynthesis. So let me clarify that a little bit. If you look at this graph, what we see here is that we have the rate of net photosynthesis versus temperature here. And in reality, net photosynthesis, which is the amount of carbon gained by leaves, is really an amalgamation of two different processes. One is gross photosynthesis, which is just the process of photosynthesis, the process of total carbon gain. But subtracted from that is respiration. Respiration is a process that occurs in mitochondria, which is basically the breaking down of sugars to release energy for the tree or the plant. And those two processes are occurring at the same time. And their sum, so basically gross photosynthesis minus respiration, equals net photosynthesis, which is the carbon available for the plant. Now, as you see, photosynthesis increases rapidly with temperature until some level at which it levels off, whereas respiration increases exponentially with temperature. So above the temperature optimum, basically respiration, which loses carbon or loses CO2, increases much faster than photosynthesis, which levels off at those higher temperatures. So at higher temperatures, basically respiratory carbon losses start outweighing uptake of carbon by photosynthesis, so we get a decline in this net. Okay. Once we get to very high temperatures, say, above 100 degrees Fahrenheit, above 35 degrees Celsius, we start seeing damage to photosynthetic membranes and even proteins sometime, and basically we start getting a breakdown of photosynthesis at that level. So low temperatures, photosynthesis and carbon gain is limited just by slowing of enzymatic reactions. At high temperatures, the balance between respiration and photosynthesis changes, and that impacts net photosynthesis until these really high temperatures at which we get actual photosynthetic damage. Now, in some species of trees have adaptations which allow them to deal with this damage that can occur at really high temperatures. And one of the mechanisms they have for dealing with that damage is that they emit a gaseous compound called isoprene. Isoprene is a volatile organic compound that's released especially in broadleaf trees that actually protects photosynthetic membranes from heat damage. And this graph shows an experiment which demonstrates the effect of isoprene. So what the researchers did in this experiment was set up leaves of oak trees in two different situations. In one of them, which is the filled circles, they had leaves of oak that were basically controlled. They were in a situation where they could produce isoprene. The hollow circles indicate leaves of oak trees that were treated with a chemical that inhibited the production of isoprene. And so what we see is that at the beginning of the experiment, and this is time on the x-axis, at the beginning of the experiment, the photosynthesis rate of these two treatments were similar. Then, during the periods here indicated by an arrow, the leaves were temporarily heated to about 40 degrees Celsius with a heat lamp. And what you see is that there's a rapid decline in photosynthesis in both of the treatments caused by that increase in respiration. But then once the heat is turned off, the control leaves returned to nearly their previous level of photosynthesis, while the leaves that were treated with an inhibitor of isoprene production basically only returned to about half of their previous level. When they again heated the leaves up to that very high stressful damaging temperature and then allowed them to cool off again, you see again the control leaves return to about that same previous level of photosynthesis, but the isoprene inhibited leaves actually were at a lower rate of photosynthesis yet again, and the same with the third treatment. So what you're seeing is that basically these leaves that kept their ability to emit isoprene were able to maintain photosynthesis even after they'd been heated to these temperatures where membrane damage can occur. And other experiments have shown that isoprene basically stabilizes photosynthetic membranes and keeps them from becoming leaky at these really high stressful temperatures. Now, we can actually see the effects of isoprene over some hardwood forests on clear sunny days. So this is a shot in the Great Smoky Mountains I took on a family vacation, and you can see there's a haze over the mountains, and that haze is actually to a large extent isoprene and other volatile organic compounds let off by the broadleaf trees in those forests. So when especially broadleaf trees are exposed to bright sunlight, they can heat up well above air temperature. So you might have, an air temperature of 85 or 90 degrees, but the leaves in those sunlit upper canopies could be experiencing temperatures well over 100. And they emit isoprene, and that allows these forests to continue photosynthesizing even when their leaves are being heated up to those damaging temperatures. So, the next time you're in the Smokies and are looking out across the mist, you have a good explanation for your kids as to exactly why that's occurring. So if you tune into the next webinar, I'll tell you why the sky is blue as well. So it's always useful information to have. Let's think a little bit about cold temperatures. So cold temperatures, as we spoke about, can inhibit photosynthesis. But once we get below freezing, we have another situation going on, and we can actually get frost damage to leaves. So when we get below freezing temperatures, you can actually get the formation of ice crystals inside leaves, and that can damage leaves and decrease their ability to photosynthesize. So this is a graph showing the effects of frost on photosynthesis of Pinus sylvestris, which is Scots pine, a northern European species. So what we're looking at here is patterns of net photosynthesis during a single day from 6 in the morning until 6 in the evening on 3 different days. So this highest curve is April 3rd, which is a day following a night that had no frost. So what you can see is we have sort of a typical bell-shaped pattern of photosynthesis through the day that we would expect on sort of a typical sunny day. However, what we see is on a couple of days, which are days after a frost event, you can see a couple of things. One, photosynthesis doesn't start until later in the morning. That's basically because the leaves are frozen and they're thawing out in the morning. So we basically get no photosynthesis until later in the morning. And then once photosynthesis starts, its maximum rate is much lower. And that's because those leaves are damaged due to that frost event. We see the same thing here on this March 14th date, which actually had an even colder night. And you can see that photosynthesis started even later in the morning and had an even lower peak in the afternoon. So, clearly frost events can inhibit the ability of evergreen trees to photosynthesize, but as we can see, those leaves do have the ability to repair themselves on this April 3rd date, which is a couple of weeks past those frost events. Those leaves have basically repaired the damage and are able to continue photosynthesizing at higher rates. So frost can have inhibitory effects through damage, but those effects can be repaired. Closer to home, Bob Teskey had some data here that is sort of similar in that he was looking at rates of leaf conductance, which is just how wide open the stomata of loblolly pine were on days following nights with particular minimum night temperatures. So what you can see is that the degree of openness of stomata, so their leaf conductance, was inhibited the colder the previous night. So on nights with minimum temperatures well below freezing, the stomata are basically inhibited and closed the following day. And so that's related to that frost damage. Basically, these leaves, because they were damaged by frost the night before, don't even open their stomata fully the following day, and that further inhibits the ability of these trees to photosynthesize. So just a couple of good pieces of data showing the effects of frost, in this case on evergreen trees. Here's another example in a southern pine. These data show rates of carbon fixation of an entire slash pine stand. So this is net CO₂ exchange on the y-axis measured with a technique called eddy covariance, which allows us to basically measure photosynthesis of an entire ecosystem. And we're plotting that net CO₂ exchange of this ecosystem versus light. So light is on the x-axis, net CO₂ exchange is on the y-axis, and the solid symbols indicate sort of typical light response, and open circles indicate response on days following a freeze event here in northern Florida. And what you can see is on days following a freeze event, again, the ability of that entire ecosystem to fix carbon was impacted. So again, just another example of the damage and the inhibition of carbon gain that can occur following a freeze event. Now, the response of species to cold and to frost is going to vary. As you might imagine, slash pine is more easily inhibited by cold temperatures and by frost than other species from more northern latitudes. So there certainly is variation among species in their ability to deal with cold and with frost. And even within a species, there are variations in their ability to deal with cold temperatures. So these are data, again, from Bob Teskey, showing on the x-axis the critical killing temperature. So at what temperature are the needles actually killed on these seedlings, and how is that related to those seedlings' growth rates? So this shows you a couple of things. First, it shows that there's variation in different loblolly pine progeny in their ability to endure cold temperatures. So what you can see is that there are some progeny, so each dot is basically a seed source that came from a different part of the loblolly pine range. You can see that there are some seed sources that are not very frost hardy. Their leaves die at temperatures below freezing, but not very far below freezing, whereas there are other progeny, probably from further north in the range, that are much more resistant to frost. But the other thing that's shown here is that in general, there's a relationship between that frost hardiness or cold hardiness and the growth rate of those seedlings. So seedlings from further south in the range that are more frost hardy tend to grow faster than seedlings that are from further northern parts of the range. And so that's an interesting relationship there. So if we think about temperature and its interaction with water, those things combined sort of start giving us some clues as to why particular species occur where they do. And we can look at this for loblolly pine, and a researcher back in the 70s did a nice bit of work looking at the loblolly pine range relative to actual evapotranspiration. And actual evapotranspiration basically looks at the amount of energy coming into the earth at different locations relative to precipitation. And what this researcher found was that on the western edge of the loblolly pine range, loblolly pine, as you might expect, was limited by low actual evapotranspiration, primarily because of dryness. Those areas on the western edge of the range are very dry. There's lots of energy input, but the actual evapotranspiration is lower because soil water limits the ability of transpiration in the west. So those are basically dry conditions. Whereas in the north, it's actually a similar low level of actual evapotranspiration, but at that point, that basically indicates lower energy input. So lower temperatures on the northern edge of the range tend to limit loblolly pine. So western edge of the range limited probably primarily by water, the northern edge of the range limited primarily by cool temperatures. And I think an important thing to keep in mind when we're thinking about temperature is that it often interacts with water or with drought. So when we're thinking about high temperatures, that often interacts with drought, and in some cases they can exacerbate each other. And some of these data are looking at pinyon pine in the west and die-offs associated with drought that have been occurring recently. And these researchers looked at predicted future temperatures and how those elevated temperatures in the future might impact drought die-off. And what they found was that these die-off events basically are likely to be exacerbated in the future, even given similar levels of drought, because temperatures will be higher. So if you think of those carbon gain graphs I showed you earlier in the presentation, basically those carbon stress situations will be exacerbated by drought and vice versa, and that those, especially in really water limited situations like the southwestern United States, may actually cause some pretty drastic mortality events. Now, we're not talking about situations as extreme in the southeastern United States, but still on the western edge of the Loblolly pine range, those are the kinds of things we probably want to start thinking about. Other issues associated with temperatures and interacting with drought is wildfire disturbance. So disturbances like wildfire can be associated with, for instance, heat waves or droughts, and those are important things to think about. Other disturbances like southern pine beetle have an important temperature element. So for instance, when we look at southern pine beetle populations, we found that winter minimum temperature is a good predictor of how far north southern pine beetle can go. And so modeling southern pine beetle populations, that sort of northern lethal temperature range is an important parameter. And the Forest Service has developed hazard maps based on temperature. So if we're thinking about temperature changes in the future, this is one thing that we'll need to think about, is how, for instance, southern pine beetle and other pest dynamics and populations might be influenced by changing temperatures. Now, when we think about responses to temperature, it's important to remember that plants do adjust, and trees can adjust to altered temperatures. So trees and plants and organisms can acclimate to changing temperatures. And acclimation just means a physiological adjustment to a change in the environment, which could include temperature or other factors. Acclimation can occur in individuals and is a relatively short time response. It's important not to confuse acclimation with adaptation. Adaptation infers a genetic change in a population associated with selection pressure. So for instance, when we think about differences in loblolly pine response to temperature of provenances from the northern part of the range compared to provenances in the southern part of the range, those are adaptation differences. Whereas we look at shorter term responses of, for instance, photosynthesis to different temperatures, those are acclimation responses. So these are some data from the University of Georgia where they looked at different families of loblolly pine grown at different temperatures. And this is a fairly complicated graph. I'm not going to go into it in great detail, but basically what they found was that at any given growth temperature at 25, 30, or 35 degrees Celsius, the trees grown in that temperature had the highest photosynthesis rate at their growth temperature. So for instance, trees grown in 25 degrees Celsius had their highest photosynthesis rate at that temperature. Trees grown at 30 degrees Celsius had their highest photosynthesis rate at that temperature, and the same at 35 degrees C. Now, there were variations in the absolute rate of photosynthesis at those different temperatures, but basically trees adjust to essentially optimize their processes at a given temperature. Now, there are limits to the degree to which adjustments can be made. So for instance, trees grown at 30 degrees Celsius weren't able to adjust their photosynthesis to have similar rates of photosynthesis as those at 25, but they were able to optimize to some extent. So it's important to remember that trees can adjust to some extent to changes in temperature. Respiration is the same way. Basically, respiration rates adjust to temperature and basically down-regulate. So the cost of doing business, which is basically what respiration is, will tend to go down as temperatures rise. So if you recall, there's that strong nonlinear increase of respiration rate to temperature, but plants are able to adjust gradually over sort of a matter of days to increase temperatures by basically down-regulating respiration a little bit. And that tends to mitigate that carbon loss associated with respiration at higher temperatures. So I just wanted to show you some of those data to emphasize that trees and other plants do have the ability to adjust to changing temperatures to some extent. I think another thing that's important to keep in mind is that especially when we're thinking about global change and changing temperatures, we do know that temperatures are going to increase in the future. That's a strong prediction, and we're pretty confident of that. And that in general, all other things being equal, increased temperatures are likely to have some negative impacts on carbon gain. But that temperature change isn't the only thing that's changing, and that carbon dioxide concentration in the atmosphere is also changing at the same time, and that those increases in carbon dioxide concentration are going to have a positive effect on photosynthesis. So these data also from the University of Georgia show in hollow bars and filled bars differences in carbon gain associated with ambient temperature in hollow and elevated temperature in solid bars. What you see is that at any given CO2 concentration, ambient plus 165 and plus 330 parts per million, in general there's a negative effect of temperature. But that as CO2 concentration goes up, the overall photosynthesis rate increases. So these increases in carbon dioxide concentration to some extent may mitigate some of the negative effects of elevated temperature. So that's perhaps good news, but it also, I think what it tells us is that we need to be aware of interacting effects of different environmental factors and how those are going to affect plants. You know, single variables don't change through time, different variables change, and those together, acting together, will impact processes of plants. We need to keep a systems view of the situation in mind. Okay, so that's some of the elements of the biology of response to temperature. I want to jump into some of the management implications of temperature and water. We'll start with temperature. There's basically not a lot we can do in terms of managing for temperature, and that's because to a large extent, temperature on a particular site is not terribly manipulatable. In most situations, we can't manipulate temperature. So a lot of what we can do in terms of temperature has to do with how we select genetics and how we deploy it. So that being said, probably the main way we can manipulate temperature is through how we manipulate the overstory. So for instance, you know, we're all familiar with Shelterwood Regeneration, which is a regeneration technique where we leave some overstory trees. And I recall from silviculture years ago when I took it, the definition of a shelterwood cut relative to a seed tree cut is that a shelterwood, by definition, modifies the understory microclimate. So a shelterwood modifies the microclimate of your regenerating understory. And in some situations, that may actually alleviate stress due to elevated temperatures. And that may be manifested as differences in water stress. So these are data from eastern Texas, which is often hot and stressful. And what these authors showed were data from 3 different regeneration cuts with different levels of residual basal areas. So we have residual basal areas of 0, 4.6, and 9.2 square meters per hectare. If you want to convert that to square feet per acre, just multiply that by about 4. So this is a residual basal area of about 36 and about 20 probably. And what you can see is that seedlings regenerated in that understory of those different cuts had varying levels of water stress. So these are measures of water potential where more negative values indicate more water stress. And what you can see is that the clear cut was the most stressful. It had the most negative values of water stress, whereas the greater overstory retention had less levels of water stress. So trees regenerating underneath the shelterwood had less water stress than trees regenerating in a clear cut. And when they looked at survival, they found similar results in that trees regenerating in the clear cut had lower levels of survival than trees with some degree of overstory retention. Now, one thing that these authors were interested in looking at was, okay, but to what extent do the overstory trees compete with these seedlings for water? So they also had a trenching treatment where they dug trenches around the seedlings to isolate them from the roots of the overstory trees. And in general, those trenching treatments made no difference. So basically what this study showed, and some others showed, was that there is a protective effect of the overstory, and that the overstory trees basically had more of a positive effect through their basically capturing of some of the incoming radiation and cooling of the microsite than any negative effect through competition they might have. So basically I think what this tells me is that shelterwood regeneration may be a viable option, especially on really harsh sites. So that's one management option to consider on harsh sites or potentially hot sites. And certainly shelterwood cuts are generally considered a natural regeneration technique, but there's no law that says you can't artificially regenerate underneath a shelterwood cut if you're interested in protecting those seedlings from heavy radiation and heat early in their life. Okay, another factor related to temperature and managing temperature is thinking about different provenances of loblolly pine growing in different parts of the range. And this map shows the natural range of loblolly pine in gray and has isolines showing the growing season length in different parts of the range. What you can see is that the growing season length in the southern part of the loblolly pine range is much, much longer than in the northern part of the range, which is what you would expect. But what that does is that starts making you ask some questions. Hey, what if I take seed from the southern portion of the range where those trees are adapted to long growing seasons and move them north, where they basically grow as if they have a longer growing season and will grow longer into the fall. You could potentially increase total growth by moving provenances north. And that certainly is true. But of course, there are risks associated with that. By doing that, you have the potential of trees growing further into the fall and actually being impacted by early season frosts. And for that reason, the Forest Service has put out seed deployment guidelines which tell us basically how we can move loblolly pine provenances around on the landscape. And they've divided the loblolly pine range up into chunks. So basically they have, moving east to west, they have 3 chunks divided by these solid lines. So one rule is you should not move loblolly pine provenances across one of these solid lines. And basically this is because in the western part of the range, we're getting into the part of the range that's temperature limit or water limited, and provenances in the west are more drought tolerant. Thinking about north-south movements, the other rule is that you should not move seed across a dotted line on the seed deployment map. So that allows us to move seed around, but not to the extent that we'd be encountering risk associated with early frost. So these maps are somewhat conservative, but when you're planting seed that's going to be in place for a couple of decades, it's not always a bad idea to be conservative. So these maps are really useful in terms of guidelines for moving seed around on the landscape. So for instance, we can move seed east to west from this part of Georgia into Mississippi as long as we don't cross that dotted line across a solid line. Now, one thing to keep in mind is that those seed deployment guidelines are based on basically minimum temperatures and are very similar to the USDA Plant Hardiness zones. And as temperatures are rising, those plant hardiness zones are changing. And it's likely that we will see revisions in those seed deployment guidelines based on changing temperatures and predicted future temperatures. So the PINEMAP project, which is a large regional climate change project in the southeastern United States, is looking at using data and predictions of temperature to help people understand how those seed deployment guidelines might change in the future as temperatures rise. I'm not going to talk about exceptions to those guidelines right now. So I think, the take-home message in terms of temperature management and genetic deployment is to choose the right seed source. So how do you do that? And the most important thing you can do is ask a lot of questions from your seedling vendor. And some of those kinds of questions are, what level of genetic improvement are these seedlings you're selling me? Are they first generation, second generation, even third generation? Each subsequent generation tends to have better selection for growth and or disease resistance. And you need to also ask, are these trees improved for growth? Are they improved for disease resistance? And for which diseases? What's the nature of the seedlings that you're selling me? Are they half sib, which means they share one common parent, the same mother? Are they full sib, or are they clone? All of those different deployment types will have different levels of performance, so you need to understand that, and there will be costs associated with that. What's the provenance? Where did the mothers and fathers of these seedlings that you're selling me evolve? And what does that mean in terms of where I can deploy this material? So your seedling vendor should be able to help you look at, for instance, those seed deployment guidelines and understand where you can plant seedlings. And I'll be frank, if you have a conversation with a vendor and they're not able or not willing to answer these questions, I would suggest that you talk to another vendor. I think most vendors would be quite interested in discussing these issues with you, and I think these are the kinds of things you need to know when you're buying seedlings that, again, you're going to plant out and leave there for 20 years. So good questions to ask your seedling vendor. So let's move on to managing for soil moisture. When we're thinking about soil moisture, we could have too much water or not enough water, and I'll talk about some elements of that in terms of managing excessive soil moisture, regeneration, density management, and so on. So it's certainly possible to have too much water. These are some data from a site in northern Florida where we're looking at efficiency, which is basically the amount of biomass produced per unit absorbed radiation, versus water balance. Across many years, where basically we had some really dry years and some really wet years. And especially early in stand development, what we found was that the really wet years were actually more stressful than the drier years that we encountered. So especially in the flatwoods of northern Florida where this study took place, these really wet conditions can be quite stressful to tree seedlings. So managing for too much water can be an important consideration on some sites. And one of the tools that we have for dealing with really wet, poorly drained sites is bedding. Bedding basically creates a raised microsite on which we can plant seedlings, and it allows us to mitigate high water tables that may exist on some sites. Bedding allows us to lift seedling roots above the zone of inundation and basically allows those roots to get oxygen. And that's especially important early in stand development where water tables tend to be high. Once the trees get established and start transpiring a lot of water, the water table tends to go down and those effects become less important. And that's sort of shown in these data. This is one of Tom Fox's students on a coastal plain site where at age 21, we're still seeing the positive effects of bedding and ditching, in this case, another drainage technique, whereas by age 33, those effects had gone away. But earlier in stand development, the effects of bedding were still apparent. Bedding has some other effects as well. It does incorporate surface soil into the rooting zone, so it can increase the organic content of soils, and it can also reduce competition to some extent. So bedding has a couple of positive effects on particular sites. And obviously it shouldn't be done everywhere, and it has a cost that you should consider relative to its benefits. So that's sort of one of our main techniques for dealing with too much water. But decreased water availability is obviously a concern, especially along the edges of the range. So a lot of the effects of limiting water are really going to be big in the western parts of the range. That tends to be the place where water balance is already marginal. So for instance, here in the drought of 2011, we can see in this part of the Loblolly pine range in eastern Texas, things were really bad. And those are the kinds of places where we really need to be thinking about these kinds of stresses and managing for them. You know, mortality can be an issue in severe droughts. It's fairly rare to see death of mature trees during drought, but it can happen along the edges of the range sometimes, or on sites where water balance is already marginal. So this is an example of birch die-off on a rocky site where the topsoil is thin and water balance is probably already marginal. So those kinds of sites or those parts of the range are where that's an issue. Now, when we're thinking about the effects of low soil moisture and drought, really the most vulnerable stage is during the seedling stage. Seedlings are very vulnerable to dry soils, especially bare root seedlings. If you think about a bare root southern pine seedling, it's frankly a miracle that we can do what we do. Basically rip these seedlings out of the nursery bed, shake the soil off their roots, jam them in a bag, pull them out and jam them in the soil, and in many cases get 99% survival. So that's pretty remarkable to me, at least. So a bare root seedling is in a tough situation. So when the soil's really dry, that can be really stressful to a bare root seedling. So we need to the extent we can, mitigate some of those risks associated with dry soil during this regeneration phase. And there's a number of things we can do. So this will be a big part of what we think about in terms of mitigating drought. That includes choosing the right species for your site. I'm not going to elaborate on that, but that's an important consideration. But these other things I'm going to talk about are really important when we're thinking about regeneration. I want to mention, I'm not going to elaborate on this, but I think you always need to keep in mind that regardless of what you do, if your planting crew doesn't do the right things in terms of keeping seedlings cool, protecting the roots from the air, keeping seedlings from drying out, all of these precautions will be for naught if basically the seedling is dead or stressed before you even put it in the ground. So that's probably job one, is to make sure you have well-trained planting crews that do the right things with your seedlings so that you can make sure that these other precautions you take actually pay off. So let's talk about some of those precautions or things you can do to hedge your bets. One of them is seedling quality, which really to a large extent is related to seedling size. David South with the Nursery Co-op has done a lot of work on initial seedling size, and he has lots of data that show that the bigger seedling you put in the ground, the bigger those trees will be even years down the road. So what you can see is the relationship between, in this case, root collar diameter of seedlings that were planted, and 4 years later, bigger seedlings had bigger volume at age 4. Okay, so bigger seedlings, higher quality seedlings tend to be bigger further down the line. An important thing here is that there's an interaction with competition. So when you controlled weeds, obviously that allowed seedlings to get bigger as well. So we'll talk a little bit more about weed control in a minute. I think another important consideration is stock type. In dry years, containerized seedlings may be a smart choice. I mentioned, all the vagaries and difficulties of being a bare root seedlings. Well, in dry years, where you're anticipating, drier than normal conditions, containerized seedlings may be a good investment. Obviously, containerized seedlings come with their own soil volume. There's less root damage because the root system is intact, and basically that little plug of moist soil may allow that seedling to survive for longer periods between rain events and ensure survival. So that's something to think about in terms of mitigating dry soils. Another issue to think about is how deep seedlings are planted. And I know that when I was in school, there was, and people were talking about planting southern pine seedlings, I always heard, plant it so that the root collar, the point where the root system started, was just below the surface, that was the proper planting depth. Well, I think there's a fair amount of data that have accumulated that suggests that, especially on upland soils and well-drained soils, you may want to plant that seedling deeper than the root collar. These are some data that Curtis Vandershoff and David South have showing survival of seedlings planted at regular depth versus at deep depth. And this is the 1-to-1 line. So if you had similar survival of deep and regular planted seedlings, they would fall along this line. And what you see is that seedlings planted at deeper depths had greater survival. So on these well-drained soils, planting deeper resulted in greater survival. This is another study from Jamie Schuler in Arkansas, and he had seedlings that he planted at 3 different depths. It's sort of at planted at that sort of common recommendation of planting just to the root collar, planting so that half of the upper part of the seedling was buried, and then planting all the way to the terminal bud. This is pretty wild to me. That's pretty deep. Okay, he was burying these things basically almost all the way, so that just the terminal bud of these loblolly pine seedling was sticking up out of the ground. Well, what do you know? That treatment had an effect. In this particular year, those seedlings that were planted all the way to the terminal bud grew several inches taller than seedlings that were planted at the root collar. Perhaps even more dramatically, he had greater survival of those half-planted or all the way buried seedlings relative to seedlings that were planted to the root collar. So, I wouldn't necessarily look at these data and say, okay, you should go ahead and tell your planting crew to bury all the seedlings all the way up to the terminal bud. But I think what this does tell us is that planting deep, especially on upland sites, is probably not a bad idea. And I would seriously consider planting getting the soil well over the root collar and maybe, up onto the needles in a lot of cases, especially on upland sites where you're anticipating drying soil. And, this makes a lot of sense. If we think about a drying soil, and this is, those old planting recommendations where they're saying you should plant right at the root collar. Well, if this is 6 inches into the ground, the upper 6 inches of soil dries out relatively rapidly. And if you plant this tree, halfway over the needles, basically what you've done is you've gotten the roots 6 inches more into the soil. And that's the real benefit, is you're getting the roots into a zone of the soil that's not quite as dynamic in terms of soil moisture, and those roots may be able to reach soil water for a longer period of time. So I think we really need to reconsider the sort of traditional seedling planting guidelines, especially on upland and droughty sites. Okay. I think another important thing to consider is when you plant. I think we sort of consider our planting season to last, well into February and March in some parts of the range. But again, I think there's a lot of data that have accumulated that tell us that given decent soil moisture at the time of planting, earlier planting pays off. Basically because the earlier that you plant in the planting season, and I'm talking about September, October, November, the earlier you plant, the longer during the winter those trees have to establish a root system, settle in, and basically be ready to go in the spring. If you plant a seedling in March or April, it basically has a couple of weeks to establish a root system into that soil before its buds burst and it starts growing new foliage that has an evaporative demand. So it really, in almost all cases, pays to plant early when there's adequate or decent soil moisture. So if you're in, September and October and you have a good rain, that's a good time to get out and go ahead and get your seedlings in the ground. Okay, another site preparation approach that can be useful on some droughty sites or heavy soiled sites is subsoiling or ripping, which basically takes a shaft and put that behind a Caterpillar tractor, put that a couple of feet into the ground and drag it. That helps to break up heavy soils. It can break the plow pan on old field sites and can allow roots to access a greater volume of soil on some sites. It can also sort of provide a little pot that can accumulate water. So on some sites, ripping or subsoiling may be a good choice. Competition control is also important on droughty sites. Competition for moisture can be very important, and there are lots of data out there that show that controlling competing vegetation both increases survival and increases height growth. So when we're thinking about Protecting against drought, there's a number of things we can do. Make sure you use good high-quality seedlings. Consider containerized seedlings. Depending on your site, you might consider ripping or subsoiling. I think planting deep is not a bad idea on most upland sites. Certainly consider planting early on most any site if the soil moisture is adequate, and on almost any site, to the degree you can, control competing vegetation. Okay. Finally, I think I want to talk very briefly about density management and water stress. And there's a couple of ways you can think about density management. One is initial planting, stocking, so how densely you plant your trees, and then thinning later on in stand development. So we're thinking about initial stocking. I think at some point in the past, people in some cases planted more densely because they anticipated mortality. And I think that has gone by the wayside and is not a good idea. Seedlings are expensive. You don't want to be overplanting in anticipation of some of them dying. And even if that were to occur, trees don't die evenly. They tend to die in patches. So really, the approach you want to be taking is to take those hedges that we just discussed and plan for good survival. Do what you can to hedge the bets so that every tree that you put out has the best chance of survival that you can manage and that you can afford. That's really the best approach in terms of stocking. Okay? In terms of density management down the line, in general, thinking about water stress, I think the best approach to managing stand density for water stress is the same as the approach we should take for sort of typical density management. So basically our goal with density management is to avoid intense inter-tree competition for resources, and that includes water. Okay, so one of I want to show you an example of why we want to manage density. So what I have here is a plot of periodic stem annual increment, so periodic stem growth in a number of stands versus stand basal area on the x-axis. And this is in metric, but what I'm showing you here is that there's a period in stand development where at a certain basal area, stand growth starts declining. And that's because of inter-tree competition. And on these stands, sort of ignore these control stands, on these stands, most of them started declining at a basal area of about 109 square feet per acre. Okay, so after that point, this is not mortality, this is just a decrease in growth rate associated with increasing stand density. So keep that 109 square feet in mind. I want to show you that in a minute. So what we want to do is avoid overstocked stands as stand development progresses, and avoid inter-tree competition, which slows growth and later on causes density-dependent mortality and causes susceptibility to insects and diseases. And one of the best tools we have for managing density is a density management diagram, which basically shows on a log-log scale average tree size versus number of trees per acre. And basically, as stands are planted at a particular density and then grow, they'll move up this diagram as they develop. So without mortality, trees per acre stays the same, but average tree size will increase. And basically, those stands will continue developing until they reach this line. So this first dashed line is 35% of maximum stand density. So there's some maximum density at which mortality will occur and stands will start self-thinning. So the goal with density management is to basically keep stands between this dashed line and this dotted line, which is basically a fully stocked stand. In a fully stocked stand, trees are growing rapidly, they're taking advantage of site resources, but they're not competing with each other, which is what occurs above this 55% of maximum stand density index. So let me give an example of that. So from our previous example, I showed you that these trees started growing more slowly at about 109 square feet per acre. That 109 square feet per acre at 605 trees per acre is a stand density index of 248. Now, Loblolly pine has a maximum stand density index of 450. That 248 is about 55% of maximum stand density index. So the guy that came up with these stand management diagrams, Reinecke, was exactly right. We start seeing that decline in growth right at this point. This is, I'm sorry, that's this line right here, that 55% of maximum stand density index. We really do want to keep stands between these two lines. And that basically tells us when stands should be thinned. So as stands develop, basically we don't want them to grow above this line. We want to thin them back to this fully stocked line and basically have stands that grow between those 2 points. So use those stand density management diagrams, and they exist for all of the southern pines, and I've given the references there for you. It's running a little late. I'm not going to talk about genetics. I just want to jump to our take-home messages. You know, I think really if we're going to worry about managing for moisture, we really need to put a lot of our efforts on the seedling or regeneration stage. Site preparation and proper seedling management are really key to survival and productivity early in stand development in dry conditions. In terms of density management, we want to use planting density to control spacing and stand development and not to anticipate mortality. So don't overplant in anticipation of mortality. And then in terms of thinning and density management, I don't think there's really anything special we do in terms of managing water, but we do want to make sure our stands don't move into an overstocked phase. If we do have dry situations, if anything, considering water basically I think makes density management a little more urgent, and we probably shouldn't let stands go into that overstock condition under really dry conditions. So if you have a choice between thinning stands on well-drained sites versus a little wetter sites, you might want to prioritize thinning in your well-drained sites. But that's basically it. I think those are the key elements we need to consider when we're managing for temperature and water. And I'll be happy to take any questions you have.
Thanks, Tim. Excellent presentation. If anyone has any questions, just type them in the chat box in your bottom left and we'll relay them. While we're waiting for questions, I'm going to go ahead and push out the web link for our surveys. You should open up a new browser. It might take just a few seconds. All right, well, if there aren't any questions, if you would give him a round of applause, there's a small smiley face icon under your name, and if you mouse over it, there's a little hand icon for digital applause.
I really appreciate everyone's attention. Please feel free to drop me an email or a call if you need if you have any questions. If you need access to any of those density management diagrams, I'll be happy to hook you up with those. Thanks very much.
Thank you, everybody.